which I conducted over the summer was on the mechanisms of hexacoordinate heme proteins binding to diatomic ligands. As humans, we all rely on proteins to survive. Proteins tend to exist in different families depending on similar structures and functions. The family of proteins we will be discussing today are the globins. You all are probably familiar with hemoglobin, right? It is found in our blood, and it transfers oxygen from our lungs to our tissues. In the recent years, scientists have discovered a new globin protein known as globin X. This is how globin X looks like. And now that is a group within this globin X protein. That heme group carries out most of the functions of globin X. Globin X is also a hexacoordinate heme protein, which I'll explain in a little bit. It contains 4 chiral rings. So that's 4 chiral rings. It contains a central iron. And now I'll explain the difference between distal and proximal histidine. So, in this globin X, it is a hexacoordinate heme protein, which contains amino acid distal histidine and amino acid proximal histidine. Distal, because it's further away from that iron atom, and proximal, because this histidine is closer to that iron atom. Now, every single protein is made up of amino acids. In this particular protein, globin X, it contains 200 amino acids. Let's focus on only one amino acid right now. Highlighted in red is amino acid cysteine. Cysteine has this unique property where it can bind to another cysteine. So, in this case, cysteine 65, the first highlighted red C, it's cysteine 65 because it's the 65th amino acid out of the 200. And then this cysteine 141, which is the 141st amino acid out of the 200, when those two bind, they form this disulfide bond that you see right here. And we think that that disulfide bond could possibly modulate the activity of the globin X protein. Now, what did we do in this lab? We measured the rate at which diatomic ligand cyanide binds to globin X. Diatomic, meaning 2 atoms, an example would be oxygen, and we use cyanide as a model for other diatomic ligands, such as oxygen, because it was easier to work with. Now, let's take a look at this diagram right here. So, remember how I explained what hexacoordinate was earlier? So, in this case, because that globin X is hexacoordinated, this distal histidine must unbind from this orange iron atom to allow cyanide to covalently bind to the iron atom instead. So in this diagram, just replace oxygen with cyanide, and that's what we did in the lab. So, what was the objective of this lab? It was to see how mutations on globin X proteins affect how quickly ligands could bind to it. Now, let's delve deep into the experiment. So, the control group of my experiment was wild-type globin X. And the variables we tested are mutating this cysteine 65. Remember, that cysteine 65 is a component of that disulfide bond. We mutated that cysteine 65 to another amino acid, alanine. And we did this because we wanted to study what effect the lack of that disulfide bond would have on ligand binding to globin X. We also mutated this distal histidine 90 to valine, another amino acid, because we wanted to explore how that hexacoordinate structure of globin X could impact ligand binding. So, by mutating that histidine to valine, we changed the structure of globin X from hexacoordinate to pentacoordinate. And we want to see how could this affect how quickly that cyanide could bind to it. Now, let's get even more into the experiment. So, you see that little cuvette right there? In this little cuvette, we first added phosphate buffer to regulate the pH of the solution. Then, we added the globin X protein. And lastly, we added cyanide. What did we do with that cuvette after? Voila, we put it in this UV vis spectroscopy machine, shown right there. Now I'm just going to briefly explain how that machine works. So, in this diagram, when light is passed through this monochromator, And onto this sample of globin X and cyanide, the sample's electrons get excited because they absorb a photon and they jump to a higher energy level. After that, the number of photons that are absorbed will be calculated by this detector. What do we get after we find the absorbance value? We use that Beer-Lambert law below, which allows us to relate the absorbance of the sample to the concentration of globin X by solving for C. Now, what did we do after that? We took that absorbance value and we graphed it over time. See, absorbance over time. Then we fit it to that equation shown there. After that, we plot the rate constant, so how quickly that cyanide bound to the globin X, as a function of cyanide concentrations. After that, we fit that to appropriate ligand binding models to derive what is known as KD. KD is just the ligand concentration at which 50% of that cyanide is bound to globin X. Now, let's move on to the results of my experiment. So right off the bat, for wild-type globin X, we see that there's a non-linear relationship between the rate constant and the cyanide concentration. This is because that distal histidine is so tightly bound to the iron, it limits how quick that cyanide could bind to the iron, thus leading to that non-linear relationship. Now, in the histidine to valine mutant, we see a linear relationship between the rate constant and the cyanide concentration. This is because now that we mutated that histidine to valine, that histidine is no longer acting as a rate-limiting factor. It is no longer limiting how quickly that cyanide could bind to that globin X. And lastly, let's get into our cysteine mutant. So, we see a linear relationship between the rate constant and the cyanide concentration. And now in this case, we possibly think that that disulfide bond I showed you earlier affects how quickly that cyanide could bind to that globin X. We think that that disulfide bond causes that distal histidine to be less tightly bound to the iron, thus allowing that cyanide to bind quicker. Now, let's get into the KD values. Just to refresh your memories, KD is just the ligand concentration at which 50% of that cyanide is bound to the globin X. So, the KD for the wild type was the highest compared to the other mutants. It was 3.7 millimolars. And because the KD was higher for the wild type than the other mutants, this indicates a decrease in affinity of globin X for cyanide. Now, the KD for the histidine mutant and the cysteine mutant was much lower than for the wild type. And this indicates an increase in globin X affinity for cyanide. Lastly, let's get into the conclusions of my lab. So, just to summarize, the H90V mutation led to an increase in globin X affinity for cyanide by a factor of 100. Likewise, the cysteine mutation led to an increase in globin X affinity for cyanide by a factor of 12. Further investigation would be needed to see how other ways that disulfide bond could possibly modulate the activity of globin X, not only in ligand binding. So now, this study provides a lot of information on the structural and ligand binding properties of other hexacoordinate globins that are not only present in fish, amphibians, and reptiles, but that are also present in every single one of us sitting in this room. Thank you. I would like to acknowledge my mentor, Dr. Miksovska, my grad student, Jonathan Sedeno Lugo, the SRI program, Dr. Etta Mills, Amy Reed, and Asla. Thank you so much for helping me prepare for this special day. Thank you.